Spring in the North Atlantic. For the past 3 months, waves generated by winter storms have mixed the surface water to depths as great as 150 meters, forming a homogeneous layer characterized by its consistent temperature, salinity, and clarity. This nutrient-rich layer and the chemical, physical, and biological changes that occur in it during the spring and early summer are the focus of JGOF's, the Joint Global Ocean Flux Study. John Mara, Chief Scientist for Leg 2.
The objectives of the Global Ocean Flux Study, which is part of the NSF's Global Geosciences Initiative, is to understand the ocean's role in the global carbon cycle. Our purpose on this cruise is a first step in that study. We wanted to try to understand a major signal in the global carbon cycle, or the ocean's role in the global carbon cycle, through the study of the spring bloom of phytoplankton, or the spring phytoplankton increase in the North Atlantic Ocean. Therefore, we designed a program, an experimental program, along with several European nations, to try to measure the flux of carbon into the ocean from the atmosphere, the flux of carbon into the particulate matter, which is primarily by marine photosynthesis, and the flux of carbon as particulate matter as those particles settle through the water column to the ocean bottom. And of course, this is extremely important given that man is now undertaking a major biogeochemical experiment by causing an increase in carbon dioxide in the atmosphere, which right now has unknown effects over the next 30 or 40 years.
The Atlantis II, a research vessel operated by Woods Hole Oceanographic Institute, prepares to depart Reykjavik, Iceland for a 28-day research cruise to study the changes occurring in late April and early May. Tons of equipment to be used by 24 international scientists on this and subsequent legs is loaded aboard. Loading goes on day and night in all kinds of weather. Lab trailers are welded on the deck. Lab equipment is set up and tied down. Equipment for processing and analyzing the samples collected is carefully secured to prevent damage in the event of rough weather. Sailing out of Reykjavik Harbor, we head south. The scientists introduce themselves and their projects to each other.
I'm Hans Stam from Horn Point Labs, and I'll be doing probably the most old-fashioned oceanography. I'll be throwing nets over the side.
We pass the surface buoy of a sediment trap deployed earlier in the season and continue south to the first station at 59 degrees north. Kurt Cochran, Terry Hammer, and Tom McKibben of Woods Hole attach submersible pumps to the winch cable at specific intervals and lower them over the side. For the next 10 hours, these pumps will move thousands of liters of water through the attached filters, which have been treated to capture thorium-234. Because this radioactive element is present in such small quantities, sampling large amounts of water is necessary to collect a detectable amount. Kurt Cochran continues.
The thorium has a chemistry that allows it to attach very strongly to particles. And because it's radioactive, we use it like a clock to determine the rates at which particles are cycled in the oceans. And fundamentally, of course, this is linked to the rates at which particles are formed in the oceans, the rates at which nutrients are taken up by the phytoplankton and converted to organic matter, particulate organic matter. So we're trying to use these radioactive tracers as clocks or tracers for the rates at which nutrients are cycled and particles are formed and decomposed in the oceans.
After completion of the pump cast, we head south to 47 North. Atlantis 2 stayed at 47 North for 2 weeks, measuring the changes in the water column over time. Of particular interest is the phytoplankton bloom, which occurs each spring when the weather patterns make for calmer seas and clear skies. Phytoplankton, microscopic plants that drift in the near-surface waters, use carbon dioxide from the water during photosynthesis. This lowers the partial pressure of carbon dioxide in the water, which allows the water to dissolve more carbon dioxide from the atmosphere. On an oceanic scale, this ocean sink of carbon dioxide and its mechanics is increasingly important due to increasing carbon dioxide concentrations in the atmosphere from pollution. In order to examine the physical and chemical changes occurring in the water column, water has to be collected from various depths and brought on board for analysis. A rosette of Niskin bottles is lowered over the side. Along with the bottles, the rosette has a CTD for measuring conductivity, which is relatable to salinity, temperature, and depth, as well as a transmissometer, which detects the amount of suspended material in the water.
Maybe 0 and 20 meters, if we can work something out like that, at least give us 2 shots in the active area up here.
Real-time data from these instruments is displayed on the computer in the lab as the rosette is lowered to its maximum depth. As it is brought back to the surface, an electrical signal is sent to the rosette, closing each bottle at each chosen depth. Once on board, small samples of water are taken from each bottle of the rosette for chemical, biological, and sediment analysis. Nadia Williams of Scripps analyzed water samples for nitrate, phosphate, and silica. The duration of the bloom is governed by the amount of nutrients available for the phytoplankton. It was thought that nitrate was the limiting nutrient. However, Nadia found silica to be depleted first, ending the bloom for diatoms, which need silica to grow. This total depletion of silica suggests the initial bloom community was dominated by diatoms, and other experiments supported this. Dan Rapetta and Carol Arnowski of Woods Hole filtered water samples from the rosette. The filter paper was then ground up, treated with acetone, and centrifuged. The liquid fraction, consisting of chlorophyll and auxiliary pigments, was put in a chromatograph, and a 3D computer display of absorption peaks versus time was shown. Analysis of this data showed a shift from an equilibrium community of coccolithophorids and diatoms to one dominated by diatoms. John Mara, Sean Chamberlain, and Carol Knudsen of Lamont Doherty measured the rate of growth of the phytoplankton community. Water was collected from various depths well before sunrise, spiked with carbon-14, and put in plastic bottles. These are put over the side on a free-floating drifter just before sunrise and allowed to drift undisturbed all day. You got it. After sunset, the drifter is retrieved, the samples collected and analyzed. John Mara continues.
And then we measure the amount of radioisotope that appears in the particulate fraction after a period of time, and that's proportional to the amount of marine photosynthesis that occurs. And this gives us the measure of plant or primary production in the ocean. And this is the basis, this is the primary carbon fixation process and the source of all the food material for the zooplankton, the fish, and the source of all the particulate matter that falls to the bottom of the ocean.
Since photosynthesis produces oxygen, increasing amounts of oxygen dissolved in the water is further indication of increasing activity by the phytoplankton. Bob Williams of Scripps measured the amount of dissolved oxygen present using the Winkler method, which precipitates an oxidized compound of manganese using the oxygen dissolved in the sample. Once the precipitate has been redissolved, potassium iodide is added, which releases the amount of iodine proportional to the oxygen in the sample. The amount of thiosulfate titrate needed to clear the solution measures the amount of iodine present, which indicates the amount of dissolved oxygen in the sample. Catherine Goyer of Woods Hole measured the pH of the rosette samples. A chamber is filled with the sample and a titrate added to normalize the pH. As with Bob's work, the changing amount of titrate is indicative of carbon dioxide depletion. Rapid growth of the phytoplankton community also results in physical changes in the water column. Wilfred Gardner and Ian Walsh of Texas A&M used a 35mm camera system to visually quantify readings from a transmissometer. Termed LAPS for Large Aggregate Photosystem, this system photographs particulate matter and aggregates as they settle through the water column towards the bottom. This movement or flux of carbon-rich particles out of the upper water column is thought to be a major contributor to maintaining the ocean's capacity to absorb additional carbon dioxide. Strobe light is used to expose the particulate matter suspended in the water. Transmissometer readings, along with conductivity, depth, and temperature, are recorded by a computer. This data and the film are retrieved once the camera is back on deck. In a related study involving particulate matter much too fine to be detected by the photosystem, Ian filters water from a rosette cast. Working in a glove box to prevent any contamination by airborne dust, the pre-weighed filters are rinsed with distilled water to remove any salts and are then put into a plastic case to be reweighed back in Texas. The amount of material is so small that even the humidity of the room where they are weighed must be the same as when the filter was initially weighed. While the laps and filtering look at sediment concentrations at a given depth and time, Craig Hunter of Moss Landing is more interested in collecting sediment at a given depth over a period of time. The particle intercept trap, PIT, was deployed as soon as we arrived at 47 North. Tubes filled with seawater made denser by the addition of additional salt and formalin are attached at specific points on the Kevlar cable. Covers on the tubes are held in place with rubber bands and lifesaver lollipop triggers. Once in the water, the candy dissolves and the covers come off. The denser water within the tubes prevents much mixing with the surrounding water. The Kevlar cable, plastic supports, and plastic tubes are used to prevent contamination of the trace metal experiments that are part of this array. The final instruments attached are a current meter and a data logger to record temperature, salinity, and the light reaching the instrument. A large buoy complete with a satellite transmitter to aid in tracking completes the operation. The pit was allowed to drift for 2 weeks while we were on station and recovered before leaving. Analysis of sediments collected in the tubes will be done on shore. Hans Dahm of Horn Point and Sigrun Jonsdottir of Stony Brook examined the actual biology of the near-surface water. Zooplankton tows were used to measure the biomass present. The nets are 1 meter in diameter and are towed slowly behind the ship to a depth of about 100 meters. Water flows through the netting and out the gut end. Material caught in the net collects in this end. Hans explains his research.
What I'm doing right now is trying to get the algae as much away, as much as possible from the animals, because we want to get carbon content of the different size fractions, and we don't want it to be contaminated with the algae. Okay, so what we have here is 2 jars that have been actually from the same tow, all right? And what we've done is separate the crustacean portion of it from the algal portion. This green stuff is all algae that we collected on our 0.2 millimeter zooplankton net. And what that means is that a lot of this phytoplankton bloom right now is not being grazed by these guys, by the crustaceans, and it's actually sinking below the euphotic zone. And it's as it sinks, you know, as it sinks, it catches on to other things, and you know it becomes what is called an aggregate. Okay, and as it catches onto other things, its density increases, its volume increases, and it starts sinking much faster. So in a sense, it's a vehicle for taking material out of the upper layers of the ocean in a way that is much faster than you would just think if you thought of the algae as individual particles. Okay.
Photosynthesis is dependent on sunlight. Daily at local noon, William Ray of JPL measured the optical characteristics of the water column. Real-time data on photosynthetically available radiation, PAR, temperature, fluorescence, and transmissivity to depths of about 100 meters is displayed and recorded. The white line on the left of the screen shows PAR decreasing to 0 at around 30 meters. The purple line shows increasing transmissivity, and the yellow line decreasing fluorescence, also at 30 meters. This indicates a concentration of phytoplankton in the upper 30 meters of the water column. Daily comparison of this data showed a shallowing of the euphotic zone as the bloom progressed. Ship operations are expensive, and the area that ships can sample is small and really limited to one point at any given time. Attempts to cover the whole North Atlantic, let alone all the world's oceans, with ships in order to collect data from multiple points at the same time would be impossible.
NASA 428, we'll have our MER in the water at 1400 and we can keep it in the water, I think, until 1430 when we've got another operation going. The euphotic zone depth today is 40 meters. The mixed layer depth from this morning's CPD cast is still 35 meters. The surface chlorophyll is, I don't have the measurements from today, but yesterday it was about 0.8 micrograms per liter. Over.
Domino pizza.
Aircraft with airborne sensor arrays can cover a much larger area. NASA 428 is an Orion P3, which is equipped to make active and passive measurements of the ocean's surface. Flying at 500 feet, the aircraft flew over the research ship, collecting data that can later be compared with that data collected on the ship. Flying out of Shannon, Ireland, NASA 428 carries a group of ocean researchers who appreciate the fact that aircraft have a port call every night. Leaving Shannon, we turn over the Shannon estuary and head for the North Atlantic. Once over water, data collection begins immediately. Sensors on the bottom of the aircraft record radiation passively emitted by the phytoplankton in the water below. Pulsed blue-green and UV lasers are used to excite the phytoplankton